Alcohol use disorder is a complex condition with a substantial genetic component. Scientists have long sought to understand how genes influence an individual's vulnerability to alcoholism. Recent breakthroughs in stem cell technology and neuroscience have opened exciting avenues for investigating the intricate interplay between genes, the brain's immune system, and alcohol exposure. This essay delves into a groundbreaking study that unveils the critical role of microglia, the brain's resident immune cells, in mediating the genetic risk for alcohol use disorder. Our brains are constantly working to maintain a delicate balance. This balance is crucial for healthy brain function. Microglia, the immune sentinels of the brain, play a vital role in this equilibrium. They act as the first line of defense against infection and injury, engulfing cellular debris and releasing signaling molecules that modulate neuronal activity. However, when their responses become excessive or prolonged, microglia can contribute to neuroinflammation and damage, potentially increasing the risk for various brain disorders, including alcohol use disorder. Understanding how genetic risk for alcohol use disorder influences microglial responses to alcohol is essential for developing effective prevention and treatment strategies. This knowledge could lead to personalized interventions tailored to an individual's genetic makeup, paving the way for a future where the devastating consequences of alcoholism can be mitigated or even prevented. Microglia, the brain's dedicated immune cells, are exquisitely sensitive to their environment. They constantly survey their surroundings, detecting and responding to any signs of trouble. Alcohol, even at relatively low doses, can perturb the brain's delicate balance, triggering microglial activation. This activation is characterized by changes in cell shape, increased production of inflammatory molecules, and enhanced phagocytic activity. Essentially, the microglia ramp up their efforts to engulf and neutralize the perceived threat. While this immune response is initially protective, chronic alcohol exposure can send microglia into overdrive, leading to a chronic state of low-grade neuroinflammation. This persistent inflammation can damage neurons and disrupt the intricate communication networks within the brain. It's like having the smoke alarm going off constantly. Eventually, the alarm itself becomes a problem. A groundbreaking study published recently has shed light on how this microglial response to alcohol differs in individuals with varying genetic risks for alcohol use disorder. The researchers used induced pluripotent stem cells derived from blood samples of individuals with high and low genetic risk for alcohol use disorder. Induced pluripotent stem cells possess a remarkable ability. They can be coaxed to differentiate into various cell types, including microglia, allowing scientists to study these cells in a dish. This innovative approach enabled the researchers to directly compare the behavior of microglia derived from individuals with high and low genetic risk for alcohol use disorder, both in the presence and absence of alcohol. This comparison revealed striking differences in how these cells responded to alcohol, providing valuable insights into the cellular and molecular underpinnings of alcohol use disorder vulnerability. The study's findings revealed a remarkable difference in how microglia from high-risk and low-risk individuals responded to alcohol. Even before any alcohol exposure, the high-risk microglia exhibited signs of heightened immune alertness. They showed increased expression of genes involved in cell division and a decreased expression of genes related to immune signaling and antigen presentation. This suggests that these cells might be primed for a more reactive state, even in the absence of any overt challenge. Upon exposure to alcohol, the differences between the two groups became even more pronounced. The high-risk microglia displayed a significantly more exaggerated shift towards an activated state characterized by a rounded shape and increased expression of genes involved in immune response and phagocytosis. It was as if these cells were on high alert, ready to overreact to any perceived threat. Intriguingly, the researchers identified a specific gene, CLEX7A, which plays a role in phagocytosis, as being significantly upregulated in the high-risk microglia after alcohol exposure. This increase was observed at both the gene and protein levels, and these cells also showed increased phagocytic activity. This finding suggests that CLEX7A might be a key player in mediating the exaggerated immune response observed in high-risk microglia. These findings have profound implications for our understanding of alcohol use disorder vulnerability. The heightened immune activation and potential for synaptic pruning in individuals with high genetic risk, driven by alcohol exposure, could contribute to a vicious cycle. This cycle might involve accelerated loss of healthy brain connectivity, potentially reinforcing problematic drinking behaviors and impairing cognitive function. Future horizons, from 3D brain models to targeted therapies.